All right, we're recording. So we're going to talk today. Oh, that's going to make me insane. Okay, let me get rid of my email so it doesn't make all that noise. Uh-oh. Kenta can't get in. Good thing I didn't close my email. Let's see. Hold it. Oh my God. See if he got, no, is he in? Who's here? No, Kenta still can't get in. All right, let me email. Kenta can't get in. Let me email him back really quick. Well, maybe not really quick. So, oh, look at me. I can't type. So now you guys are watching me. All right, your heads are over. Oh, there we go. Goat. All right. Okay. Oh, was that him? No. Oh, that's Sarkis. Okay. All right, we're back. Okay. So today, my friends, right, we're going to make some PCR primers and in some of the documents that I posted the rules. I know reading is annoying. So let's go over the rules really quick before we start actually designing. So hopefully you have your sequence because we're going to design primers today. And remember, it's not like you're going to design primers and then I'm going to check to see if you're right, because there are no primers designed to these sequences, right? These are sequences that we pulled from the genome. Nobody else has ever cloned them that we know of, at least it's not published. That's not in the database. So this is all us. We generally want our primers to be 18 to 25 nucleotides long. So our primers are going to be complementary to their target, which is genomic DNA, right, our putative promoter. And we need them to be big enough so that there's enough homology for them to sit down, but not too big that makes that cumbersome or that those sequences have secondary structure. So what do I mean by secondary structure? Secondary structure? What's primary structure? That's just the sequence of nucleotides. Secondary structure would be any internal hydrogen bonding. If we think about pieces of DNA or any nucleic acid, anytime A's are near G's, right? G's, A's are near T's. Sorry, I've already lost my mind. Or G's are anywhere near C's, we get hydrogen bonding. It's just chemistry. There's no thinking about it. There's no, ooh, should I hydrogen bond? Is that my complement? No, it's just if they're close to each other, it happens. So if there's a string of DNA where we have, I'm just going to make it really dumb. These, right, this string of nucleotides, we're going to say it's a single strand of DNA. We just hit it with a lot of heat or enzymes to break up those, you know, the double helix, and it's separate. These A's are going to want to flip on itself to those T's. So this guy is going to end up looking like that. And right, these here are the phosphodiester bonds between Each one, just like they would be here, I just didn't show them, right? They're all connected together by those phosphodiester bonds. And then what happens in here? Hydrogen bonding. So that piece of DNA is going to fold on itself. So if we get back to why we give a crap about that, except that it's nerdy fun, is that the bigger our piece of DNA, the more likely that kind of secondary structure might happen. So it has to be big enough to sit down so it's homologous only to our target, right? If you think about what's in the genome, a gazillion, billion, trillion, I don't know how many nucleotides. What are the odds of your little piece, if you made 10 base pair nucleotide primer, what are the odds that that would only sit where we want it to sit and not on some other chromosome and some other gene? It's gonna, of course, it's gonna sit on some other gene. It's gonna be close enough. 9 out of 10 base pairs will make something sit. 8 out of 10, maybe. So if we make it 18 and we have it, it's then less likely that all 18 in that order, or 17 out of that 18, are in some other gene in that same exact sequence in the right orientation to do that hydrogen bonding. Right? So we don't want it too small. So we don't really want it smaller than 18 because then it might sit everywhere. We don't really want it too bigger than 25 because then we have a lot more opportunities for that kind of baloney. So that's the ideal size. Doesn't mean you can't go a little smaller or a little bigger, but we're going to try to stick to the rules.
So we don't want secondary structures?
No. So that's the other thing that the computer will help us look for and we'd want to look for. You don't want, that was 0.4 or something like that. You don't want strings of the same nucleotides. Right? So if you have a bunch of A's all together, or a bunch of T's, or a bunch of G's, or a bunch of C's, that causes some secondary weirdness aside from complementary. And then you don't want repeating sequences because those can cause secondary structure. And then you definitely don't want, you know, the N to have 3 A's, a bunch, you know, say there's 10 nucleotides in between, right? We'll say 10N, and then T's at the end. That's going to make that guy flip over, right, on himself. He's going to want to do that much more than he's going to want to sit on a piece of genomic DNA. So we throw a lot of primers into our mix. So yes, some are going to squeeze on each other, some are going to base pair to other things, but the majority we want to sit on our template. So we're going to always have some of this kind of stuff. We just want to make sure we also get some priming off of our template.
How many primers do we put in?
So we're going to make, so we'll talk in a second just about the whole point of PCR to make sure you guys understand, but we're going to be making 2 primers. So we're going to make, well, we're going to make one, and then we're going to ask the computer to make the other one for us. So we'll get to that in just a second. So what else do we want for, if we're making 1 primer, right? We want it to be between 18 and 25 nucleotides. So that's our goal. And then we want it to be between 40 and 65% GC rich. What do we mean by GC rich? That just means the number of G's and C's in that string of nucleotides. So why do we want that? We want that because I drew these like this for a reason. Between A's and T's, there are 2 hydrogen bonds. So we could go back and look at the stupid structures and look at those guys, but there's 2 hydrogen bonds between A's and T's, and there's 3 hydrogen bonds between G's and C's. So G's and C's hold on to each other a little bit tighter, and so they're more likely to stick and stay stuck without falling off. So too many A's and T's in your primers, it makes PCR not work as well. And too many, if you get up too high, over 65% GC, it sticks too well. And so then when you hit it with heat to remove them again in the process of PCR, they don't come off very well. So we want sort of the happy medium. And really, you know, our really, really happy medium is like 50%. But sometimes it's really hard to hit it right on the head. Your sequence is your sequence. So for us, we're going to have our design, we're going to want really close to a specific area. We're not going to have a lot of options. So we're going to try to fit these parameters, aiming for 50%-ish or more. Like we'd rather have it a little more than 50% rather than less, if you had a choice. And then we definitely want a G or a C to anchor the 3' end of our primer. And we want that, again, based on the rules of how physiology works, right? PCR is just DNA replication in a test tube. All it is. We're just making it happen, right? Real DNA replication happens in your cells when it's in S phase, supposed to happen. And what do we know about the 3' end of a primer in DNA replication? Anyone? Anyone? Okay, I'll tell you. DNA polymerase only polymerizes to 3' OH. That's where it adds the next nucleotide. That's how you know what direction replication is going to go. Okay, so if you think back to 360, or if you didn't have 360 yet, or you don't remember, or whoever taught it didn't teach you anything, I will post a DNA replication video. I didn't want to post it ahead of time because I didn't want to confuse people. And there are the PCR videos that I posted that like don't go into super great detail. I could not find my super good detailed PCR already recorded, so I was going to make one for you, but then I didn't. So now we're doing it. So the 3' end, regardless of all that baloney, the 3' end is the guy that's right there. That's where polymerase is going to grab and start adding more nucleotides to amplify your DNA, to do PCR, polymerase chain reaction. We are polymerizing a chain of nucleotides in a reaction. So we want that guy to have this really nice, strong interaction, those 3 hydrogen bonds there. If we can, we will anchor the 5' end also. But we definitely want a G or a C on the 3' end of our primer. Okay, I'm gonna do one for you guys so that you can see what the heck I'm talking about before you have to try it. Okay, so if this is completely foreign, you didn't have it, you don't remember it, you don't know what I'm talking about, don't panic. We're gonna get there, we'll all get together at some point. If you've got this, you're good, just hold tight, relax, and wait for the next slide. Okay, so those are the big 3 rules. I guess I could make. And then like our little question, our number 4 was our secondary structure stuff. So we don't want repeats. We don't want strings of the same nucleotides. We don't want like GA, GA, GA in a row. And that just gives primers issues. Okay, lots of A's and T's, strings of A's, strings of T's. Too many G's in a row is a problem. Too many C's in a row, right? And so in order to hit that 50%, that means you got to be about half and half. So if you have 7 G's in a row, you're already pushing towards way too many G's and C's. If the rest of that area is 50-50, you're going to push yourself way beyond that 65%. And remember, we're kind of aiming for 50, but we're giving ourselves this little wiggle room for that. And same with these guys, right? We really don't want to go over 25, but occasionally we can go under 18. It's just not such a great idea. Okay. So, hello, oops, I hit the wrong button. Okay, so in order to understand how to design a primer, we have to understand at least the basics of what PCR does, right? So we're doing research, you have to understand what you're doing. So we need to be able to think so that you're not just pipetting clear liquids into tubes, getting results. If they work, great, you don't need to think about it. If you get what you're supposed to get, boom, you can move on without thinking. you don't get what you expect to get, you get something a different size, or you don't get anything, then you need to understand the process in order to change something. Just repeating exactly what you did, unless you remember going, oh crap, I know I didn't put enzyme in the tube because it was still in the freezer. Ooh, didn't open the freezer door yesterday. Right? Like, and that's super easy to do. There's lots of steps. But if you think you did everything, you have your checklist on your protocol and you checked it all off and it didn't work, repeating the exact same thing is pointless. It's not going to work again. Right? Just keep doing the same thing over and over is not going to change anything. So you need to be able to change something. Well, what do you change? If you have no idea what's going on in that tube or what's happening, how the heck are you supposed to change anything? Right? Then you're just a technician. You're just somebody that somebody else tells what to do and you pipette it, which you can train anybody, right? You don't need an education for that. You have to be able to read directions, so you have to be able to read, right? But we could have middle school students doing that, right? As long as you can read and follow directions, you're not too squirrely, right? But that's not what research is. Research is all about thinking. So we need to understand some PCR. So, and I get it, it's not, even if you had it in class and 12 people explained it to you, it's not, nothing's the same as actually doing it. And designing primers actually helps you understand PCR better, which is why I make our brain do it rather than just asking a computer to do everything for us. So there is some method to my madness. So if we pretend like, here's your chromosome, right? It's double-stranded DNA. And we, right, we all had probably a different chromosome, but we looked in those sequences, right? We saw a gazillion sequences. And so we'll pretend like, you know, a little part of it is your gene, right? That's where we found it, right? And then right up here, that's the first ATG. And then remember, we copied about 1,000 base pairs upstream, or 5' of the gene. That's our putative promoter. It's double-stranded DNA, right? Sequence of double-stranded DNA. So we're going to pretend that now we're going to take that and we're going to make, we're only going to look at one piece, and we're kind of like going like this. So now this little tiny piece is now a bigger piece. So now I'll draw the same baloney here just to keep us on track. Here's the gene, right? That's the first ATG, right? This will save to here is 1,000 base pairs, you know, 5' going that way. And so that's our promoter. We know there's another strand there, but I'm not going to draw it because it gets too confusing. So we're just pretending like we're on the strand where this side is 5' and that side's 3'. So that's exactly like what we copied into our Word file. We copied 5' to 3', whatever that strand was. And we remember we could see our ATG. Some of us, okay, I'm gonna label this guy, right? So what's this stupid thing? Right, from here to here is like 5' UTR. That was the purple guy. And then the rest, this stuff was all white. Right, that's the genomic DNA where we assume our promoter is sitting. And so remember, some of us had an intron in here. You might have had an intron, right? Doesn't matter. What we want, can you even read that now? What we want is we want from the 5' UTR closest to your ATG, we want to make a primer. And we want him going that way. So we want its 3' end to be here. Its 5' end to be here. And that works because our template strand, the one you're looking at on your sequence, goes from 5' to 3'. And anytime we have any kind of hydrogen bonding complementary, has to be anti-parallel, right? That if these are our 3' ends and my elbows are the 5' ends, right? That's how double strands of DNA stick together. That's how an RNA to a DNA sticks together, an RNA to an RNA sticks together, right? We can never do this, which I can't even bend my arms, or that, right? We have to always be anti-parallel. So that works, And then we also want somewhere up here to get enough space. So like we're gonna make between 500 and 1,000 base pair PCR product. Okay. So this primer up here can be there, he can sit here, he can sit right at the edge. There's lots of places where we can make that 5' primer. And this guy has to be 5' to 3' in that direction to make sure it goes with the arrow, which means it's not complementary to the sequence you see in your file. It's complementary to the sequence that we didn't see. So our missing, right, if we, you know, I said I wasn't gonna draw in our other strand. If I draw in the other strand now, like that, right, this is its 5' end, and that's its 3' end. Right, this one's 5' to 3'. So the dotted line is our other strand, and then you see if our primer's 5 to 3, this is 3 to 5, then this one is complementary to our ghost strand that we don't see. That makes sense? Okay. At least anti-parallel by the rules, and they have to obey the rules. So that's what I was yammering about on one of the other videos, where we want our primers facing each other. We want our thumbs to be the 3' ends, because we want polymerase to go this way. We don't want it to go that way. We don't want it to go that way. We don't want it to go that way. That's not going to make PCR. So if I change colors, just so maybe that will be helpful. I wish they gave me more than 3 automatically. I'm sure there's some default I could do. Right, so we're gonna pretend like this is the primer we're using. And this is the primer we're using. And this guy's gonna polymerize going this way. And this guy's gonna polymerize going that way. And then we're gonna do it again. And we're gonna polymerize, and we're gonna polymerize. And if our primers sit on our new strands, then they're short. If our primers sit on our template strand, then they're gonna be long again. They're gonna follow that whole chromosome. So what do I mean by that? Without drawing all that other stuff, just PCR in general, and I'll draw our pretend other strand. If we're doing PCR, I'm not going to do 5' to 3' since I don't want to get all confused and do it wrong because I have to think. So we're just going to pretend they're sitting on the correct strand in the correct orientations, right? And I'll make our polymerized new strand be red. So in PCR, we throw the genomic DNA in, we throw these primers, we throw the nucleotides, the individual guys that need to come in, and we throw in a polymerase. So essentially we're doing everything just like DNA replication in a cell, except we're doing it in a test tube. And we say, go. Polymerase, go. And it grabs the 3' end, which is the arrow end, and it starts adding nucleotides complementary to whatever template sitting there. And so it's just gonna go until it gets tired and falls off, right? Chromosomes are 1 billion base pairs long. I don't really know how long they are, but they're way longer than one polymerase could handle, right? So think about if you're going for a run, it's it's not easy to run a block, but it's much easier to run a block, but you might get tired and stop at 3 miles. Even if the road still goes another 20, doesn't mean you're tired, you're done. So that's the same as polymerase. But polymerase doesn't know you want it to stop right around here. How the hell would it know? It's just doing its job. It's just polymerizing, right? So it just follows along its template till it poops out. Same with this one. He's going this way. Okay. So then in PCR, all the old strand and new strand are complementary base paired and they're hydrogen bonded. In replication, we have DNA gyrase that pulls them apart for replication. In PCR, we just heat the crap out of it. If you hit it to about boiling, hydrogen bonds break. And so that all these are single-stranded. So now what we'd have after we hit it with some heat is we'll have this, our original template, our original ghost template strand. Oops. Right, those are the originals. We have this guy. And that guy. Okay. So now we throw in a whole bunch more primers. And now this, we're just going to pretend that another green primer is going to sit on the strand, right? Because it's original green guy. This guy fell off because we heated it up. So he's not attached anymore. He's, you know, floating around on his own right here with his strand, right? He's a big old strand. So now this guy's going to sit there. And that's the same as goes for the other guy. Now he's going to sit here instead of just where he was, right? Because now that guy's this guy. But the beauty, here's where the beauty and the nerdiness comes in. This guy can also sit on this strand, right? It's complementary. If it was made like something else, then it's still going to be complementary. And same with this strand right here. Yes, yes. And so now when we say polymerase go, this one, complementary base pairs. This one, complementary base pairs, but its template's done right there, right? That's the end of the primer. Primer and new strand are attached, right? That's a phosphodiester bond. So it can, this is where, here is where DNA polymerase falls off. There's nowhere else to complementary. It can't just put nucleotides out of, pull nucleotides out of its ass and put them in random orders. It needs that template, so it falls off. This one keeps going because its template kept going. This one that went back to its regular strand keeps going. Oops, I lied. Help. I did a bad. Watch, I'm gonna fix it. This one did pull it right out of his ass and keep polymerizing. Sorry. He has to end right there. And then we heat it again. Oh God, it's gonna get complicated. Oh, Nelly, come on guys, stay all together. I'm just gonna try to grab him and move him. I don't know if I can grab him all at one. Oh, he's not moving. Go. Oh, I lost their strands. Oh wait, I forgot to give this guy his strand. Right, he keeps going because his template doesn't end. And then when we do, we heat these up and separate them all, right? We're gonna get that same thing. This time, this guy can sit here, right? Somebody's sitting here, somebody's sitting here, somebody's sitting here, somebody's sitting here. And now we make this piece as short. This piece still goes long and follows that guy. This piece is short, this piece is short. And I don't remember where else I've had a guy that should be short or long. But see, because I don't have it all mapped out at a time. But you can see, then when we heat these and these guys fall apart, we're gonna get a lot more of this little piece each time because there's only one template and that never changes. We make that one big long strand on him. Right? But every round we make a couple more of these small ones, and then we can, on those two, we can make two more, which is 4, and then we can make 2 more of each one of those, which is 8, and 2 more of those, which is 16, 2 more of those is 32, 2 more of those is 64, and so we get exponential amplification of this piece, right? These guys and these guys, and then we still get a few of these long ones once in a while. But that's how PCR works. So when they show it in books, and I'm sure even in the animation that, or the graphics I showed from the textbook are still like that, they show you this. They show you, here's your template. Right, here's our, ghost strand. And they say, here's your product. here's your primers, and he goes to here, and he goes to here, and then now this one sits here, and this one sits here, and you know, this one sits here, and that one sits there, and then you just go back and forth, and then he's to here, and to here, and to here, and to here. Woohoo, we get amplification. Well, yeah, that's totally true if this was your entire chromosome and you put primers on the ends of a chromosome. If you're doing PCR somewhere in the middle, it doesn't know to stop. Like, really, this template is like this, right? We're taking a little tiny piece out of a giant chromosome. So there's no way it wouldn't know to stop there. It would keep going. It only stops if it primed off of one of the newer strands that is the end of the strand. So let that soak in your head, because I know that's a crazy concept, right? And we do need to know it, but we don't absolutely have to know it to design primers. It does help us understand about our 5' primer.
That doesn't happen until the 3rd cycle, right?
Yeah, I mean, that template's in there the whole time. So if you have 25 cycles, you're going to have 25. That's a great question. You're going to have 25 of these long strands. But you might have, you know, I don't know, I'm going to make it up, 50,000 of the short strands. So when you look at DNA, we don't see it by molecule, right? We look at it on a gel, and we stain it with a fluorescent dye that stains DNA. And you have to see, I don't know how many molecules, I'd say at least 5,000 molecules to see it on a gel. So we're never going to see 25 molecules. So it looks like that doesn't happen, but it's really in your tube. We just don't see it. So that's why in classes they try to simplify it and just not show you those ends and just show you, look, it amplifies DNA, see? Without being complicated. But it ends up confusing people, because when we talk about making our 5' primer, you have lots of choices. You don't have to make it on that very end that you copied, because we don't care. We want about 500 base pairs close to that first ATG or close to your 5' UTR. And so knowing that there's lots more sequence, there's another gazillion sequences farther up. Like, let's say you can't find a 5' primer that works, you can just copy more DNA and make it bigger. We need our 3' primer where we want it, because we want to get all of the DNA down by the 5' UTR. So I'm going to pull in, I think, if I remember how, my sequence, so that I can show you what the heck I'm talking about on an actual sequence. Now I gotta log in, I guess. See, I should have all had this done ahead of time, but I had to eat, and I was in my other class till 11:30, so. Let me in. It's gonna make me do it again. Oh, let me in already, god damn it. All right, so we are, where are we? Hello, am I blind? Oh, yes, I am. Are you kidding me? Where are my sequence? How did I do this last time? I'm like, it's odd. Talk amongst yourselves. What the hell? That's bizarre. It makes me. Hmm. Um. Wow, I have no idea. Let me log in again.
Wait, so are the green arrows DNA polymerase?
No, the green arrows are the primers.
Okay.
And then DNA polymerase grabs the 3' end of those guys.
Okay.
Yeah, I'm trying to get my. What? I did this. I swear I did this on Wednesday. I don't know where I got the file from if it's not actually in my files. Let's see. I'm going to put it in there now so I can use it. Hello, hello. So have your sequence. And it's much easier if you have an iPad or have it printed out, which I know is annoying when we're not, like normally we'd be, you know, in a classroom and we'd just print it on the computer, on the printer, and then, you know, design primers just like that way. And I know at home, like I don't have a printer that's working right now, but I can put it onto my, theoretically, I can get this onto my iPad to do the actual, because when you're complementary base pairing, it's a lot harder. The hell? Oh, I'm in the wrong one. To like eyeball it and have the sequence on the computer and be writing something down on a piece of paper without being able to line it up. So I feel for the problem. Okay. Thank you very much. Okay, so here is a promoter sequence that I pulled. It's from the human V-pre-B promoter. That doesn't matter. And this, let me get it bigger so I can show you guys. So this would be that 5' end. So this is the ATG, right? And so then if, we'll pretend like this is all the purple sequence. We're just gonna make it conveniently this far, right? And then the ATG, and then everything that was down here, that's into our gene. That would be what the pink stuff would be, right? This would have.
Professor, I can't see your screen.
Oh, sorry, thank you. Nobody can. I shut you guys out. Ah, that's not helpful. Okay, wait. Thank you. Okay. Oh crap, it's gonna make me do this again. Okay, now can you see? Hang on, let me move it over. Are the heads in the way? Okay. All right, so here's my sequence, right? Sorry, I'll go back. This was the 5' end, the farthest up on the page, right? And I just copy and pasted. This would be all the white from that NCBI thing. And then this went into the purple, the 5' UTR, and it went right to my ATG, right? It would be an M, and then these would be pink, and it would have all the amino acids under it. Now I know some of you guys have an intron somewhere in here, so I'll do that little just in case. So, like I know some of you guys had green intron in here. So if you have no intron, if your sequence got right up to your ATG, then you're going to want to try to make your primer somewhere close to that ATG. Ideally, we'd want it sitting exactly next to it. Get everything of that 5' UTR, but not including the ATG. So you don't want to anchor, you don't want to have that this G be in it, or this T or this A. So you'd want to start with this guy or any of the ones in here. Ideally, we want our primer somewhere right here. That's not a lot of sequences to choose from to get all, remember the 3 rules, the 4 rules to make that happen, which is why we have wiggle room. And then we're going to ask the computer to make the one that comes up here, because this side we want to be really close because we want this 5' UTR. If we think about it, that would be the +1 of transcription, right? Transcription makes all of the RNA. So the UTR, the 5' UTR, the purple, is the 5' untranslated region of the RNA. So it's part of transcription. So if you have this intron, this green right here, then you're going to want to start your primers on this side, somewhere pretty darn close to where that intron is, as close as you can get, right? Because we don't, even though this intron, all this sequence is part of the RNA, because introns only get spliced out after RNA is made or during the making, our transcription start site for this, we'd say, was here. Actually, let me, it's not really that, oh, that's not what I wanted. I suck at this. Okay, we are gonna use that. Okay, let's say this, I'm gonna highlight it. And I'm gonna change, and I'm gonna say our 5' UTR, oh, it's a little bit too fat, goes to that C right there. Okay, sorry, I changed it. So this is right here is the +1 of transcription. Are we okay with that? I know I just changed my story and made it more confusing. We'll pretend like that was where the 5' UTR ends. Okay. Because I want to explain why we think the promoter is right upstream of that. So if we think about transcription, and again, this goes back to whatever we learned in 107 and 360 or wherever you took it at community college. DNA polymerase, or sorry, RNA polymerase, RNA polymerase is the guy that does the complementary base pairing. And so that guy is going to use this first C, is the first C of RNA. So it had to be complementary base paired to something on the DNA, right, our other strand, in order, and then each one was polymerized. So we're going in that direction all the way through that intron if we had one, all the way through, all the way through, all the way through, until we get through that 3' UTR, right? That's how we make RNA. How does RNA polymerase know where to sit? How does it know that's the +1 of transcription? That's the promoter sequence. Yeah. Right? The promoter sequence's job is to tell polymerase where to sit. We don't want polymerase sitting here or down here and missing half of the gene, or sitting, you know, somewhere way down there. We want it to sit right here. And the way that that ordeal works, and it's going to be an estimation. Here we'll say this is polymerase. He's a big guy. And he sees essentially this sequence. And then he has a bunch of friends that help him out, some friends. And essentially this end is his catalytic activity. We'll pretend like this is his enzymatic activity. And so he grabs the DNA, or his friends grab the DNA and help him get there. And then he starts complementary base pairing. And then this whole mass just chunks down the DNA making RNA. So the sequence right here, right off of that 5' UTR, whether you have an intron or you're going all the way here, is where most of the action should be. now we're showing this, you know, then all we need is this. Well, sometimes sequences up here through these sequences actually help out that blob and some of his friends get together and make transcription go. But generally, we don't need this sequence out here isn't impacting how that guy sits or what he does. So what we want to PCR is all of this, because sometimes he has friends that sit down here to help guide him. So we don't want to miss those friends, the proteins that bind to those sequences. So we want the 5' UTR. And then we probably only need really, sequences up to here. So we want all this sequence to be PCR'd. So we want, I should have made that a different color. We want to make a primer going this way, so 3' end here, and a primer somewhere that sits up here going that way. We don't need to make our primer right there. We can, but we don't have to have all that sequence. We can make a primer that's right there, or right here, or right here, or right here. We have hundreds of places where our forward primer, that we like to call it, I like to call it the 5' primer, but, and this I like to call the 3' primer, but most people call it reverse. So we really want to reverse right here or right here if you have an intron. So my intron friends, you're going to be right here before your green intron for your reverse. If you don't have an intron, you're going to try to get right up against that ATG. But then our forward, we have lots of options, because most of our action, according to everything in the literature, everything that's been published on promoters for the last 30 years, says 95% of promoter action is within the first 300 base pairs. But we're going to pull at least 500 just to make sure we're not one of the 5% that has some action at minus 400. But we don't have to take all 1,000. We can, but there's generally nothing fun up here happening to help drive transcription. So that's why I wanted you guys to understand what was 5' UTR, where the coding region is, what's genomic sequence, so that you can make an educated decision about where you can put your primers, right? Because in research, your PI, your advisor, they shouldn't design the primers for you and say, here, pipette. Because again, like we said, I could do that to a middle school student or anybody that walks in off the street to give me a hand, right? I want you to understand what you're doing so that you can, well, and save me some time. I can't be designing everybody's primers, right? So that you guys can say, okay, I know why I need it here, not just because Dr. Malone said so, but because it makes sense. And if these like, so that gives us this really narrow range, right? Just like, I want it to be within about 50 base pairs of that. We don't want to miss any of this good stuff. It could totally screw up. You might get a nice PCR product, and then you're going to clone it. We just, you know, Harout asked me about, you know, or was that Harout? I don't know. Whoever asked me about restriction enzymes on the end of primers and how are we going to clone. We're going to clone into that intermediate vector that we don't really care about, but it sucks up DNA nice and easy. And then we're going to subclone into a vector that we can look at, does this promoter work? Is it functional? That's the experiment. PCR is not our research experiment. It's just a step towards our actual experiment. Our actual experiment is, does this sequence drive transcription? Is it a promoter? So if you get a really nice, let's say you're like, hey, I found some really, I found some rockin' primers that are awesome, and my forward primer is like right here, and my reverse primer is right here, and I get this kick-ass PCR product. when you clone that and test it for transcription, it's not going to be a promoter. So you just spent a semester of work doing PCR. Okay, yay, you learned how to do PCR, you can put it on your CV. But if you want to continue in your research, you have to start all over and make primers again and go through all of that and then test that again. So this is important because it's essential to the function, right? So if I want to be, if I want to be a good instructor, which I do, but I don't know why it won't let me, like, I used to be able to like squeeze it. Oh no, I don't want that. Oh, there we go. And it all goes together. Yay, why wouldn't it do that on that other page? Okay, sorry.
We're just looking for the core promoter, right?
Yeah, I mean, we're looking for the core promoter and the proximal promoter. And textbooks kind of talk about it differently than how people in the field in publications talk about it. The core promoter technically is just where the basal transcription factors sit with RNA polymerase. And so that would really be only about 50 base pairs. The proximal promoter includes other friends, other transcription factors that bind sequences that are usually like 200 base pairs upstream. So the core basal transcription is usually pretty low and sometimes undetectable in the laboratory setting. So if we just PCR just this little piece right here, this should be enough for RNA polymerase to sit on. We won't see good transcription. We really need like this line and this line and probably this line to hear all of this sequence to really get transcription to work, because other transcription factors help those core like TF2Ds and those and TATA binding protein and the RNA polymerase. And so we try to take up to here just in case. Sometimes there's fancy, transcription factors helping out at around 500 base pairs away. Usually within 200, 300, but sometimes 500. And again, we don't want to not take that stuff now and wait till we test it to find out we should have taken it, right? Okay, so does that make sense?
Yeah, thank you.
Thank you. So what's our hypothesis? That our promoter is upstream, which means 5', right, that way, of 5' UTR, and within 500 base pairs or so. Right, that would be our hypothesis at this point. So we're going to test it. We're going to PCR out this bad boy, throw it in a vector, and then another vector, and we're going to test it. Is it a promoter? And then we can then look at the nitty-gritty of sequences. Here. Do we really need these guys? We're going to cut it off. Does it still work? If it does, then this stuff up here was crap. We'll keep removing sequences until we lose activity, and then we can go back and do site-directed mutagenesis and see, is it this GACA CTG sequence that's important? Which transcription factors might sit here? We can ask a computer to tell us, and it'll tell us. And then we can see what transcription factors are sitting there. Is the 5' UTR important? Is this important? Is that important? We can look and find out exactly how this gene is being regulated. But we have to start with, right, making ourselves, well, first thing we have to start with is making this primer. And we can get back to the big picture, and I think some of those other videos has my big picture in it. You can always go back and watch that again, because again, like when you watch it, you're with me, and then sometimes later when you think about it, it makes your head spin around a little bit and you gotta think about it some more. So, now that I have, now that I have all this crap on here, how am I gonna do this? All right, let's try grabbing my sequence again. Am I still logged in? No, I gotta do stupid shit. Okay, yeah, we go. All right, there we go. That's easier. Okay, so now I'm going to talk about, after an hour of talking, I'm going to tell you what we're doing today. Okay, so I'm going to just make this big as I can, like that. Because we don't care about that, right? Remember, here's my old ATG guy. And I always label our polarities, so that helps me remember. And remember, for this strand here, that's the 3' end. So anything on this side would be the 5' end, right? We're looking at, and that's just like how you copied your stuff too. And again, if you're an intron person, you're gonna be going to the left of your little intron. If you're not an intron person, then you are going to be making your primer as close as you can to your ATG. So if I draw, I'm gonna try right up against it. What the hell? So here's what I want my primer to be. And I want it to be 5' to 3', right? I definitely want this end to be the 3' end because I want it to go this way. If I made my primer with the 3' end over here, it'll PCR in that direction into the coding sequence. I don't want that, right? We want this all the way up. So I'm gonna, and because I'm looking at this strand, this is 3', and I can even make a 5' here. That helps me, right? Every end over here is the 5', right? Every single nucleotide is a 3' end, a 5' end, and a 3' end. And so I'm going to, this guy, since it's anti-parallel to our target, we're going to make it complementary. So we want it to sit on what we see on this strand. So what am I going to have to make here? I'm going to have to make this be a G because that sits on C, correct? Yeah. And then it would be a G, whoops. And then A goes with T, C, G, C, A, G, A, C, right? We're like off schedule, but A, T, G, C. Uh-oh. Let's go that, guys. That, okay. A, G, A, G, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. A. Right, wait a minute. G. Oh crap. A, G. Sorry, because I'm writing too big. This C is that C? Yes, so then that C. I would have screwed up. This has to be a C here. And then T, C. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So I can make mine 20. I could cut it at this C also. That'd be 18. But since there's a C right here, I'll try 20. So this guy is 20. Did I do that right? Do I have to check myself here? Okay, let's check. Because if you screw it up and you buy this primer and you do the PCR, it's not gonna work. So it's not like, oh, you're gonna lose points. It's not about points here. It's about, right, getting stuff to work. All right, so let me just check this guy. If you wanna try on yours, you can either write out your sequence on a piece of paper, unless you can print your sequence so that you can do your complementary base pairing without trying to look at your computer and write on a piece of paper. If you have better spatial than I do and you can do that, then by all means, you don't have to write it out or print it out, like that's up to you. But do that now so that you can do the next step with me when we go and ask the computer what would be a good 5' primer to go with this. And is our primer good or does it stink, right? We're gonna do our eyeball, is it good or stink? But then we're gonna also ask a computer. So I'm gonna check my sequence. You guys try to do some complementary base pairing. see. Okay, so my sequence is good. All right. So we're just making the reverse primer because it's going that way. Since we read left to right, we call the ones going this way forward and the ones going against how we read reverse. So let's see. Hmm? So I just highlighted the sequence that mine is complementary to try to make it a little easier to see. And then, so if you're rewatching this video at a later date to generate a primer to check yours, you'll have it how I did it. And you see how it's easy to make a mistake because I made one. Luckily, I caught it. Okay, and then I am going to do something else. Okay. I'm going to stop this share so I can open our other site. So hopefully you guys don't need that in the meantime. Okay. Yes, yes, yes. And pull up our other site that we're going to use. Did I pull it up? Uh-oh, ding dong. What happened? Who's ding-donging me? Okay. Why don't I have it in here? Oh, there we go. Okay, so then after we get our primer made, or if you're having issues, we can also work on it later. But I just want to show you guys the next step so that it's on our recording. And then you can do that later if you don't have your primer finished yet. So that you can see if you have your primer finished, or even if it's not as long as it's supposed to be or what you think it is. See, I already forgot. I was going to do one more thing. Oh boy, you guys, my brain. Let's see if mine obey the rules first. Right, remember our rules? Oh, darn it. Come on, there we go. Because I just, winged it, right? Of course, I did this last time. That's why I forgot. Go away. Okay. So here's my primer. Oh, move this guy over. So you guys can keep designing and just kind of half listen to me. And it'll be on the recording. You can always watch it again. We talked about the rules. We wanted it to be between 18. Oh, that's not helpful. Between 18 and 25. And so mine was 20 nucleotides, 20 nukes. So that's within our range. And then let's look at GC rich. How do we figure that out? Oh, we count. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 GCs out of 20. So I can tell already that's more than 50%, because I'm a math genius. No, because I'm not a math genius, but I'm gonna use my calculator to figure out the exact percentage. So that is exactly 60%. So it's 60% GC rich because I have 12 GCs out of 20 total. And that's how we do percents, right? So that fits my criteria. What was our third one? We had to be GC rich. Oh, we got an anchor. And what's the anchor? It is a C. That's the 3' end. We wanted to anchor G or C because it's got the 3 hydrogen bonds. So 1, 2, 3 works. And then all I'd have to do is look and see if we have a bunch of repeats that might make it fold. I don't see any strings of A's or T's or G's or C's. CTC, GAG's not so great. That could flip on itself, but it's so small and there's nobody in between to help it. There's no hinge in there, like they're right next to each other. So it would actually be, if you think about stereochemistry, it would be hard for those guys to bend on their little phosphodiester bond to hydrogen bond each other. So this guy could be the hinge, and then it could go like, so we might have blah, blah, blah. And then this, it would have to skip that one to go T, C here. And so that's not super likely, right? Because that's not really going to fit this extra G that he can't really hydrogen bond to this guy because he's too close. So that's probably not going to be a deal breaker, but we'll ask the computer later. Okay, so I made those 3 criteria so we can eyeball those up front just to make sure you hit the big 3, and then we'll still ask the computer. So if you can't do that now and you just want to try the program, you don't have to do any of that right now in your head. We'll just do it. You can do that later. Okay. So I gotta switch my stuff around. So we're gonna go to NCBI, our favorite website, and we're gonna just put in Google NCBI primer blast, and we're gonna get this page, and then we're gonna click it to open it up. Oh, I need my iPad. And then this is going to be where we're going to ask it to find primers. So you can use this in a lot of different ways. What we're going to do first is we have our own reverse primer because we want that guy to sit right where we want it. We don't want to give it any options. Okay, so this would be where we put in the sequence of that primer we just made. And we need to put in our reference sequence. So that's the sequence of our promoter. Right? If we don't have that in there, then it's not going to know what the heck we're asking it. So first, we're going to go back to our promoter sequence, and we're just going to copy paste into here. And then we just want to make sure that we, like in our original file, that we don't have any like, I wonder if it would show me. We don't want any, like the hard returns at the end, right? We want it to just be, I don't know how to look at that. But anyways, if it's wrong, if you have spaces or something weird in there, it'll tell you. The system won't work. So if you know you returned on every end of that sequence, remove those. Okay, and then here's where we put in our 3' primer. But if we look carefully, we have to write it in the 5' to 3' direction. Now with any kind of nucleotide, nucleic acids, RNA, DNA, whatever, we're always assuming left to right is 5' to 3'. Okay, so if I typed it in as I read that primer, as I just wrote it on my iPad, it would be wrong because I'd be typing in 3' to 5'. So what I should do, and I will do, oh, look, I can squeeze. Okay, I'm squeezing that one. I'm going to send us back to, oh, why can't I just jump back? Sending us back to the iPad, of course. It's nice I went forward because then it tells you why I'm doing it, blah, blah, blah. But I know it's annoying because now I'm going back to the other page. Okay, so what I'm saying, what I'm talking about is this guy that I drew, right, is left to right 3' to 5', because, you know, I had to complementary it this way. So if I want, so now I need to change that sequence to be in the 5' to 3' direction, reading left to right. So all that means is I take this, the 5', and I write it here, and I say if I'm reading right to left, that first sequence is a G, and then a G, and then a T, and then a G, and then a C, A, G, A, C, A, T, G. Oh, I'm running out of room. C, I just write smaller, A, G, A, G, C, T, C. I wrote too big on the other side. So I didn't just change. You can't just go, you can't just say, okay, I'll label this side 5' and this side 3' and order that, because then it's gonna be wrong. And it'll sit on the other strand, but it's gonna go in the wrong direction. So you're not gonna get amplification of PCR. You're gonna get a bunch of long stuff we don't care about. So I just took, right, there's the 5', so this is this G, that's G, T, G, C, A, G, A, right, all the way through. So it's that same exact sequence. It's the same primer. It's just written 5' to 3', okay? And my head, I'm very spatial, but in a weird way, like I have a hard time turning things around and seeing them. So, you know, because DNA is printed out like that, it doesn't mean it's not sitting like this inside a cell, right? It can be any direction. So just because we drew it like this doesn't mean this thing can't turn around and bind to the DNA. But my brain sometimes gets crazy. So in case you have a crazy brain like mine, don't panic. It's not abnormal. Well, it may be abnormal, but other people have it too. Okay. So if you couldn't do all of that in this little time, you can always watch this again and try it on your own when you have more time. But I rewrote, so I designed my primer, I checked it, I rewrote it as 5' to 3', and now I got to type it in here. And so now, so all these different switches, you have to be really careful you don't accidentally type or write the wrong thing, or again, it'll turn out to be wrong, and then PCR won't work. So so mine ended up being GGT, GCA, GCA, GAC, ATG, CAG, AGC, TC. Okay. Right? Yes. And so in the real world, we usually read to each other to type stuff in, or have somebody else look at it and you read to them to make sure you don't order it wrong. Primers aren't expensive, but it's annoying because you don't know until you don't get a PCR product, and that could be weeks later, and then it just sucks. You'd rather have somebody catch, you know, a typo or an error before you do it. So we always help each other and double check. Okay, and so then the PCR product size, we don't want our minimum to only be 70 base pairs. We want our minimum to be about 500, right? That was our goal. We could make it smaller. If we get nothing back and the computer can't find a decent primer in that big region, max I'm gonna leave as 1,000 because I don't even have more than 1,000 base pairs in here, so that doesn't really matter. Number of primers to return, yeah, I'm hoping I get 1. I don't think we're gonna get 10, but we'll ask for 10, you know. And if we get more than that, then we might make some of these parameters more stringent to make them better, but we're gonna leave it at default right now just so that we get a primer to come back. Melting temperatures, right? We wanted, the optimal is about 60. This kind of correlates with GC richness, but there's a formula for this in the PCR procedures PDF if you want to actually do yours by hand to see. There's a formula in there to do melting temperature. I just use the GC because it's the cheap and easy way. And we don't want their melting temperature to be very different. We want it to be really similar. So if we did get back tons of primers, we might change this to be 2 degrees or 1 degree to get rid of the ones that aren't as good. We don't need to look at 10. We only want 1 pair, and we want the best one. So we can always make this more difficult and get rid of some primers. But we're going to leave it here just in case that's all we get. And we can make this a 4-degree difference if we have to get more primers if we got 0. We don't care about exons and introns. This would be only if we're PCRing off a coding region. We're not. We're PCRing off of the non-coding, the upstream, the genomic. Blah, blah, blah. Transcripts, we wanted to look at the Homo sapiens because that's the gene came from human. Okay, so this also looks for mismatches, which means, is it going to sit on other chromosomes and other genes? And so we're going to leave this as the default right now. But again, you can always make it more stringent so that it has less chance of, if you get lots of primers, you can make it more stringent so that it won't sit on anything else and see how many primers you get back. Blah, blah, blah. We don't need splice variants. We're not doing RNA. We're going to show it in a new window. Highlighted in yellow. Okay, we didn't change anything. And then we're going to say go. So we put in the 3'. We're looking for a 5'. And we're going to see what our program, and it'll tell you, it usually comes back in about a minute. Usually not much more than that. So if you're making this happen or whenever you do, take a screenshot of your results. I don't have any place to upload stuff yet because I can't get my act together. I've got too many other Canvas classes adding and subtracting and making rubrics and this and that. And I had, life issues. 31 seconds have passed. So it should come back pretty soon, and then we'll see what it gave us. And again, if you're doing it along with me, yay. If you can't, if you didn't get everything together, you can always do it again later. That was 51 seconds. Oh my gosh, I lied and said a minute. It's gonna be longer. Come on, you could do it. Oh. I think Wednesday it only took us 51 seconds. What is happening?
Mine is at 66 now.
What is happening?
Oh, it worked. Yay. Okay.
Oh, you beat me. See, we should have had a competition. Anybody else? And does anybody else have a question? Okay, so I think I gave you guys a little better explanations than Wednesday. So I better tell the Wednesday people to watch today's. Although I don't know. Watch Wednesday, you can compare and contrast.
I had a question.
Sure.
So when we copy the primer into like the big box, do you want, like if I had an intron in between, I would just copy everything but the intron?
So just copy everything to the left of the intron. So you don't want to skip it and then take that other sequence. You want to just what's up 5' upstream.
Okay.
Into that box. Yeah, it's a great question.
I wouldn't take that.
I'm sorry?
So after the intron, I wouldn't copy.
No, you don't need that in there because we don't want anything to go on there because we're going to want the chunk and we don't want that intron in our way. Okay, so here's my results. Oh, is it not going to let me see? Oh, there we go. Okay, so this is the little arrow for our 3' primer, the reverse primer that we put in. So it's all sitting exactly where, the same, they should be. And then it gave us 7 different primers, right? All sitting in different places. Okay, so again, right, that was, we told it could sit out there. We wanted minimally 500 base pairs, but it had up to that 1,000. And then when we scroll down, it'll tell us about these primer pairs. Okay, and so our reverse primer is our primer, so it should be the exact same sequence for every single one. And then the 5' is the one that the computer generated for us. It tells us how long it is. It gives it its melting temperature and its GC rich. I'm not sure what these numbers, if high numbers are good or low, or these are high or low, but I'll look that up because I just can't remember off the top of my head. But we're most interested in melting temperatures to get them as similar as possible. So like these are pretty close, but this one's a little closer. That one's not, that's 58, that's a 60, 61, right? That one's a 58, this one's 59, right? So, and then we also think, well, we have this choice of 7, what should I pick? Do we want the biggest? Do we want the smallest? So because we have choices, we could go through and say we want our melting temperature more similar. The more similar your melting temperature is, the more likely both of your primers will do well under whatever parameters we put in the PCR machine. So the PCR machine is going to heat it up to separate strands. It's going to cool it down to an annealing temperature, which is when annealing just means primer sitting on template. And then it's going to raise the temperature a little bit where polymerase is the most happy, where it has the best catalytic activity. So not so high that your primer won't fall off, right? But then you can get polymerase to start going. So each primer has its own annealing temperature based on its melting temperature. And so if they're way different, you only get to pick one annealing temperature. So you want to pick the one between both of your primers. So the closer they are in melting temperature, the easier it'll be to find the annealing temperature that works for both and allows you to get PCR, right? If only one of your primers sits, you're going to get a bunch of those long strands, and that's it. The other one works, you're going to get a bunch of long strands. So it doesn't help if your annealing temperature is kick-ass for one and doesn't work at all for the other. So those melting temperatures need to be close together. And the default says 3 degrees, which probably means that's most will work with that difference. But if we can get them closer, we'd pick the closer ones. So I like 60, 62, and 61, 37 better than 58 or 59. Right? This is 60.02. Oh, this guy still wins. And then I should look up what self-complementary means. Like, is it better to have these numbers or, oops, sorry, like these numbers with my 60, 62, or these numbers, right, with my
I looked that up, the self-complementary. It
Right.
says that a score of lower than 10 is pretty much ideal because that's just the likelihood of the primer to bind to itself. So under 10 is fine.
Okay, so like, yeah, so the lower the better, but there's not a big, these are both 4. What about 3'? Same thing?
Yeah, 3', same thing.
Okay, so 0, right? So this 0 guy is better than 2, but again, probably both would work. So at that point, it's like, well, which do you like better? Right, or you just randomly pick. But you'd want to save this file, which is super annoying that I haven't figured out. Maybe one of you little youngsters who are good at stuff, there should be like download this as a PDF. It's 2020 for Pete's sake, people. So that we'd have all of this stuff so you don't have to rerun your primer to go back and remember. Right, so I mean, we can take screenshots and save them, like copy and paste in a Word, but why don't they just have download these results? And maybe they do, and I just don't know where it is, but we'll want to save it because just because the computer says these are good and you say, woohoo, I'm picking this set, it still might not work. And so then we might have to go back and try a different set. So instead of retyping in all our crap to pick, we just go back to our screenshot or our save file and say, okay, this guy didn't work. Where's the next one? Which, oh, I like this one. This one had a pretty close last time. Let's go with that one. Rather than, I mean, you can always just retype it in and regenerate it. It took, well, mine took almost 2 minutes, but it's just easier to have, right? Then you don't have to do that. The other thing that we can do, if let's say you got back only like 2 pairs, so you got back 2 pairs and both were like the maximum melting temperature apart, that 3 degrees apart. And this said they kind of stunk, right? So a 7 is worse than a 4, right? If under 10 is good, I would assume the lower the better then. You know, let's say your self-complementaries on, you know, each of those is like at 10 or 11. And you're like, and both are equally stinky, according to the computer. In that case, because primers aren't that expensive, and we only have one 3' primer, we might just order both of those 5' primers. And then we'll just have 2 reactions for our PCR machine, right? We'll have forward 1 and our reverse, and then forward number 2 and our reverse. And that doubles our chance of getting a product. Now, if both work, we just use one of the products. And then if one or the other works, we're like, yay, we didn't have to do the experiment, wait for the gel, spend a week or however long it would take between classes to get your result, and then go, oh, it didn't work, I need to order the other primer, wait for that to come in, reset it up, blah, blah, blah. But if you have a whole bunch and all of them look pretty good, we're just gonna get one set. Like, we don't need to go crazy and have everybody setting up 15, because that's how it goes. Like, either all of them work or none of them work. So that's always an option if your primers are not ideal and none of them are really good. But if they look pretty darn good, then we're just gonna, you know, we would just get one set for each person. And so if your reverse is totally crap, so like, or you get back 2 or 1 and it's total crap, and you look at these numbers, like this, you're self-complementary and you're designed by your brain, we would go back and redesign that one and try again, right? You can move it over. It doesn't have to sit right where the anal retentive Malone wants it to be. It can be over 5 base pairs or 10 base pairs. We just don't want it over 50 base pairs. We want it close, but we'd rather have it a little farther away, lose a little bit of your 5' UTR, than to not have it work at all and spend our life doing PCR and testing primers. Because that's not our experiment. This is just generating reagents for our experiment. And nerdy fun. Does that make sense? Does anybody have a question? Okay. It's 1:30, so it's been an hour and a half. I think you guys have had enough of me for today. So if you're not near this and you're still designing primers, that's all good. This takes time. We only have an hour together a week. I do have office hours Monday morning, 9 to 11. I have my Zoom on. I'm ready to help. I have my other class. It's all at the same time, but you know, I'm doing the Biol495, which is the non-majors, and they almost never come. They might come just to say hi, but you know, they're not biology majors. If they don't understand something, they're like, whatever, it's fine. Just let me out of this class. So I generally have lots of time during that chunk. If you're in class during that time chunk, shoot me an email. We can set up a Zoom at another time to help get you, you know, back on our page. You can always, what was I gonna say? You can always watch either the videos again and follow along. Just keep hitting pause while you're doing your thing. If you have questions, I see my good friend Rabia posted her email in our group chat. Does she want? Yes. So Rabia has worked in my lab for several years and she's awesome. So happy to help you guys out. If I'm unavailable or you don't want to talk to me, that's okay. I totally get it. I'm not offended. So that's resources. And again, this is research. You're allowed to work together. You're allowed to help each other. Like, as long as it's not like when I told my Wednesday people when I think, or maybe it was you guys, when I was in college, I had a lab partner who I just did everything and she followed along because she was a lost soul and that was sort of like my community service. But she didn't learn anything. And so if you're doing research, you really should want to learn. But if you're, you know, you're really behind or swamped one week, you know, lean on your lab mates and then you catch up on the understanding later. Or you know, you work with them and you work through it, and they're not going to make your primers for you, but they're going to be your cheerleader and your guide. Like, ooh, remember the 3 rules, or no, you're going the wrong way, or that sort of thing. So you're totally allowed to work together with anybody, as long as you feel like you're getting it, right? And they can do examples for you. They could, you know, you can form a little group. You could do whatever you need to. So we're not working in our own little bubble. I'm not going to give you guys a test on how to make primers, right? I posted some of those other little things about primers just to help you make sure you're with the program to make this easier for you, and to see, you know, where everybody is, and hope we're on the right page so I can give anybody more help. But yeah, definitely help each other, work together. It's like a team. It just kind of stinks that we're a team, not all around in lab, you know, laughing and joking around. We're like little boxes on a computer. Okay. So feel free, email me, email anybody else, watch all the videos, and I will post a little video, which I don't have ready yet, but I will, because it just takes so long to process. Like, good Lord. I'm going to go back quick just to give you a little preview. What I ignored up here was we can actually ask them to make primers for us, and we can set parameters for the range, meaning we want this reverse primer to only be cover these last 40 base pairs. So it can't try to make our reverse primer too far away. And then it will make a forward primer. We can make that 1,000 to 500 or whatever. So I'll make a little video for that. And then you guys can do that on your own too, and see if your primer you made is as good or better than what the computer came up with. And so we'll do, are we as good as Primer Blast? It's okay if we're not, because we're not a computer. But I'm thinking you guys are gonna beat the computer. So I'll make a tutorial for that so you guys can try that on your own whenever. Okay? Otherwise, love you guys. Thanks for being here. And email me or come see me in office hours.
Professor, can you stay for like 5 minutes? I just have a couple questions.
You bet. I'm staying. Okay, everybody
Okay.
else gets to go. Or if anybody else wants to stay, they can.
Bye, Dr. Malone.
Bye,
Dr. Malone.
guys.
Thank you.
You're welcome.
Okay, I feel like I did my, hold on. Where is it? The promoter sequence upload. I feel like I did it wrong.
Okay.
So, can I
the
just share my screen and then
Yeah,
like,
yeah. Hang on, let me get out of mine. Oh, yeah. I know, it's just such a long day that I didn't want to force people to have to stay, because I know it's been a while. Let me make sure it lets you. Advanced. What?
okay, I just gotta join on my computer.
Okay, I'm trying to say yes. Where does it? Is it letting you share? Hang on.
No, it says host disabled participant screen sharing.
Oh, it's under. Is it under participant? I just went under share to try to find it.